Assalamualaikum. I am here from Conceptual Medicos, and today in this lecture we will be talking about the antihistamines. So before going to the discussion of the antihistamine, first we should know about the histamine. So histamine is a biogenic amine which is present in many animals and plants and also in the venom and stinging organisms. And the human being, they are synthesized from an amino acid known as histidine by the process of decarboxylation to form histamine. This decarboxylation reaction is performed by this enzyme known as histidine decarboxylase. After formation, mostly they are stored in the storage granules of the mast cells, and upon activation by the antigen antibody reaction or some other stimulus, they secrete those histamines to perform their function. Mostly in the inflammatory reactions or in the allergic reactions. They are metabolized by the monoamine oxidase, which is responsible for other metabolism of the monoamines like the norepinephrine and the dopamine, and also by the diamine oxidase. The major product on metabolism of the histamine is imidazole acetic acid. So we can measure the level of histamine by measuring the level of imidazole acetic acid in the urine. Coming towards the receptors of the histamine, so there are five types of receptors of the histamines from H1 to H5. H1 and H2 being the most important and mainly located on the many organs. Note that histamine has no clinical use. Coming towards the H1 receptors, so most effect of the histamine are mediated by the H1 receptors. Note that they are GQ coupled, means they will increase the intracellular level of the inositol triphosphate and diacylglycerol, which in turn will increase the inside calcium level. Due to this increase in the calcium level, there will be contraction of the smooth muscles, so there will be bronchoconstriction. They will also cause the contraction of the endothelial cells. Let's suppose this is endothelium, this is a capillary, and these are the endothelial cells. So they will cause the contraction of the endothelial cells like this, and will promote the increase of the interendothelial gap, so will increase the permeability. They will also cause vasodilation, but note that this is not due to the effect of the contraction due to this mechanism. Basically, they are NO mediated. That's why it causes vasodilation. The second type of receptor is the H2 receptors. Note that they are not GQ coupled, but they are GS coupled, means it will increase the level of the adenylate cyclase, which will increase the level of the cyclic AMP, then the protein kinases, and then they will cause the phosphorylation of many channels. They are mostly present on the parietal cells and on the heart. So on the parietal cells, they causes increase in the gastric acid secretion. So that's why we can give anti-H2 and the peptic ulcer. They are also present on the heart and increases the SA nodal activity. The H3 receptor are mostly the presynaptic, present on the presynaptic nerve endings, which are GI coupled, and they are auto-inhibitory receptors of the histamine release. Let's suppose this is a neuron and this is H3 receptors, and from here the histamine is released. So it will cause on this H3 receptors and will cause the inhibition of the histamine release. So they causes auto-inhibition. Moreover, the things will be discussed in the next part of the video.